fact, we do have opportunities if someone's interested in pursuing research, getting 199 credit. And I will probably be even advertising an undergraduate job. So again, if you're interested, you can also contact me. Right, actually I have to remember that part. Right. I discussed it with Diego recently, but I gotta remember it. And anyway, okay, here we go, share screen. Wait a minute, that looks like, oh, here we go. That must be it. Share screen, and I'm gonna try to go here. Okay, and then I still have to, let's see, I still have to do something here. Display settings. Oop, that's always very small. Let me try that. There we go. Okay, and you got, wait, let me, I also have to move a toolbar if I can out of the way. That thing's always in the way. You guys don't see it, but I do. All righty, let's see. Okay, yeah, here we are. So we're interested in how plants respond to climate change. And, you know, there are many reasons that this is important. One of them is that we live on an oxygen-free planet. By the way, can you all hear me well?
Yes.
Okay, great. You know, we live on an oxygen-rich planet because of photosynthesis. We live in a CO2 low planet because of photosynthesis, meaning that the temperatures are manageable. And also in terms of food and agriculture, it all comes from plants. Even if you eat animals or fish, it all ends up coming from plants and photosynthesis. And so that's very important. In fact, we just released a report written with some undergraduate students in my lab. We just published that, in which we explain that agriculture is probably the industry that's going to be most impacted by climate change. Now, it's not only agriculture, it's ecology, and it's also things like forest fires. I mean, you know, we've had fires in California. You might have heard of the bushfires in Australia, and it's increasing around the world. So how plants respond to climate change, and, you know, is very important. Let me see if I can get a pointer. Oh, here we go. Yeah. Oh, wait, actually, let me go back again. Where is this? Activate a pointer, see if that works. Oh, there we go, laser pointer. Okay, you guys can see this, right?
Yeah.
Okay, good. All right. Okay. So in this slide, what we basically see is that if we think of agriculture yield on the planet, it's about 21% of what it could be optimally if all those plants received enough water and nutrients and all that good stuff. So there's a lot of room for improvement. And that's important because we live on a planet with a growing world population, with a growing quality of life. And we don't want to cut down more forests to grow our plants. So these negative losses here, that's what we want to cut into. And the major losses shown in red are from so-called abiotic stresses like drought, water, salinity, cold, and so on. Now, today's subject is on climate change, and why is that important? Well, hang on, let me just, why is this working? Okay, something is slow here. Okay, well, I'll just continue. What we're experiencing and what we've been experiencing in during the Industrial Revolution, but particularly in the last 50 years and less, is a very steep increase in the atmospheric CO2 concentration. And right now, the CO2 concentration is already 50% higher than pre-industrial revolution. And there is that slide, right? So we're seeing how this is rising. Now, carbon dioxide, you know, we worry about it because it's a greenhouse gas and it traps heat on the planet. And having 50% higher and going even higher is a big deal. On the other hand, plants like carbon dioxide. Plants use carbon dioxide as a nutrient, and that's kind of shown in this slide here. This redwood tree that's typical of the Pacific Northwest in California or in the Sequoia National Forest, the mass of this redwood tree is largely what? What is it? Question for you guys. Unmute and participate. Where do you think that mass comes from in this tree? That's just the bottom of the tree, obviously.
Comes from carbon?
Yeah, most of this is carbon, right? And it's fixed out of the atmosphere by photosynthesis. And photosynthesis is incredibly effective because even though we're worried about the CO2 concentration in the atmosphere, and justifiably so, the concentration is 0.04% of the atmosphere, but CO2 is a heat trapping gas, so it's important. So you might say, if there's more CO2 in the atmosphere, shouldn't plants be growing better? And there have been satellite studies of this. This is from a study that actually showed globally, because there's more CO2 in the atmosphere, there was a greening, an increased greening in the planet in many regions. But that only went until the year 2000. And this basically is a satellite approach that looks at that sort of green cover or leaf cover of the whole planet. And this means increases in greening, and this means decrease. So you can see until the year 2000, there was an increase in photosynthesis of land plants on the planet. However, was it enough to capture all the carbon? No, because otherwise we wouldn't see that steep increase. It captures about not quite half of the carbon we're pumping into the atmosphere, and that equation could get worse or better depending on how we deal with energy. But here also, like the predictions, right? So right now we're in the year 2021 and we're at about 415 parts per million CO2. If we don't do anything about this, we're going to be on this trajectory. And if we do a whole lot, which could be quite difficult, we'll be on this trajectory. But any trajectory in between is going to be helpful because they determine how much the globe will increase in temperature. And you might not be able to read this here, but the blue curve says 8.5 at the end. Why? Because the predicted average global temperature increase is 8.5 centigrade or Celsius. Well, to some Americans, that might not sound like a lot. To you, I know you know that. But then if you say, hey, 8.5 Celsius is 15 degrees Fahrenheit, that's a pretty big difference. And some of these others you see here also have substantial increases. So in the worst case scenario, okay, let me see here, freezing again. So anyway, in the worst case scenario, what's going to happen is that the planet is going to get very hot. You know, when you have a day that's it's like 80 degrees, fine. Suddenly that day might be 95 degrees. If you have a day that's 95 degrees, being 110 isn't a very pleasant thing. So yeah, I don't know why this thing is freezing. I don't even see. Let me try this again. Now we have this. It's funny, I was teaching all last quarter. I didn't have this problem, but apparently it's happening now. Maybe I'm gonna have to throw out one of my slides there. Can I go down here? It doesn't matter. Okay, well, there is that next slide that we already talked about. So this is like the prediction for an average 15 degrees Fahrenheit warmer planet. And you know that you've probably heard that on the poles, the heating is particularly strong, but also this is in centigrade, but also in all other regions, right? Now, if we can hold CO2 down, we're going to be looking at a much better situation. This is for a 2.6 degrees centigrade average increase in temperature. Right now, we're, I think, already at about 1.2 above. And you might think these small changes are not that big a deal, but if our average temperature across the globe would sink by 2 degrees centigrade, we would have ice covering most of Canada and Northern America. So it makes a big difference. Anyway, today we want to also talk about drought because this is obviously linked when you have increased heat, drought, forest fires, and so on. Oh boy. Yeah, there's something wrong with this. I don't know if I should reboot my computer or what's going to happen here, because this is not funny. Do you guys think, should I try a quick reboot and log back on? I mean, right
Julian,
now, yeah,
do you want to send me the slides and maybe it'll work better from my end?
I can try that. Right now, I can't even get out of this mode because it's frozen. But yeah, I'll try that. But right now I'm stuck here. I'd have to turn off the computer. Wow, then there's something else that happens. Okay. Yeah, I'll send them to you. I have to, actually, they might be, the file might be too large for that. Okay, well, why do plants need so much water? You know, plants are very sensitive to water. About 70% of fresh water used on the planet is used in agriculture for plants. And in dry states like California, 80%, in other even more dry regions, 90% of fresh water is used for plants. And the reason for this is exactly what we talked about earlier. Plants breathe in carbon dioxide to fix the carbon and release oxygen. And they breathe in carbon dioxide. Does someone know how plants take in carbon dioxide?
Through stomata.
Through stomata, exactly. And the key point here is that for every carbon dioxide molecule that's fixed by photosynthesis, plants lose 300 water molecules. So plants use a lot of water globally. And I wish, you know, I can turn my computer off to reboot it, but it seems to kind of be stuck in this mode here. So yeah, I have a feeling I might just turn off my computer and log back on. I'll see if I can. Okay, should we take a 3-minute break? 4-minute break? I'm gonna try that because I don't know what else to do with this.
We can try that.
And I'll just do this here. Go through the whole procedure again here. Let's see here.
If it freezes again, one thing I've seen is turning off your camera makes it a little bit easier for your computer to run like other programs.
Yeah, I haven't had that problem before, but maybe I'll not turn on the pointer. You can see my cursor here. No, you can't. I'm going to turn on the pointer. I mean, usually this is always work when I'm teaching. I don't know what's. I have a feeling that there's one slide in this thing that's not. Oh wow. Okay. Anyway. So as I mentioned earlier, for every carbon dioxide molecule that goes into the plant, plants lose 30 water molecules. Now, that's actually getting worse because as the temperature is increasing globally, there's something that's called the vapor pressure deficit. And this actually is derived from thermodynamics, but as you increase, but you don't need thermodynamics. As you increase the temperature, the evaporation of water increases out of leaves. So this problem is actually getting worse. And that's why this greening of the planet that we saw until the year 2000 has stopped. And if anything, you know, well, I guess it's kind of plateauing now, but it could decline. So, you know, what's predicted globally are losses in agricultural production on all of the continents. And these are examples for different countries. So, you know, all this sounds pretty negative, and I don't want you guys thinking it's only negative because, and I thought I'd introduce a little quiz here so we can have some fun. And so let me ask the following question. Wow, am I already stuck again here? Okay. So this is kind of a question you guys should unmute, you know, forget about chat right now, but if you unmute, So what do you think the ratio of the total solar power that's reaching the surface of the Earth is compared to total human power use? Anybody throw out a number? We're just guessing here.
0.1%.
0.1%, meaning in which, like, what's more?
Oh, the human power use is less than the solar power reaching the surface of the Earth.
So that's about 1,000 to 1. Yeah.
Yeah.
Anybody else?
1,000,000 to 1.
Say what?
1,000,000 to 1.
That'd be nice. Yeah, no, but at least you guys are thinking more positive than some other people. Yep. And another, how about one more guess?
I'm guessing the solar power is less. So maybe about 1 to 1,000.
That solar power is less. Yeah. Okay. All right. Well, why don't we go to an answer here? And I guess what I'm trying to show you here is that there are a lot of opportunities for us here. So the total amount of power that reaches the surface of the planet Earth is about 90,000 terawatts. Okay. All human power use, whether it's cars, heating, air conditioning, electricity, lighting, airplanes, ships, everything on the planet is 20 terawatts. So there's about 4,500 times more solar energy reaching the planet than all human power use. So we can solve this problem. It's just a matter of will, and as we see, politics play a role. But keep this in mind. It's really, I mean, there's a huge industry and huge job opportunities in the future just by exploiting this. And you know, that's solar power. We haven't even talked about wind power, which is actually cheaper today still to produce. And what about gravitational power from the tides, from the moon, right? Pulling waters back and forth, and so on and so forth, and geothermal and nuclear. All of those are carbon free powers. Of course, nuclear presently is more expensive than people are concerned. But what I'm trying to say is the problem can be solved. And plants also play a role because they are the best and the cheapest decarbonization mechanism, probably trees right now. So all of this is important. But if we're going to grow trees to decarbonize, help pull carbon out of the atmosphere, we're going to need to make these trees stress resistant. So we need to understand stress resistance. So I wanted to mention one thing that's kind of beneficial along these lines: elevated CO2. So these are the stomatal pores through which plants lose 95% of their water, and they breathe in CO2. Well, it turns out elevated CO2 is also a signal that reduces stomatal apertures. And that's a good thing in a way, because if there's more CO2 in the atmosphere, you can actually have less of an opening and lose less water. So rather than losing 300, you could lose less water molecules. And there's a second effect in some plants that if you grow at elevated CO2, you get a reduction in stomatal development. So we're interested in how this works. And when we started out, this is more or less what we knew. That's only a little over 10 years ago. We've learned a whole lot, and I'm not going to give a lecture on this. I'm going to tell you, how do we screen for mutants? Because some of this then we get into bioinformatics, but that's only one example that I'm going to show you today. So first of all, how do we screen for mutants? Well, we use highly sensitive infrared thermal imaging cameras. These cameras were originally developed for like military or type purposes, and they've gotten really good. So here's a plant. It's an Arabidopsis plant, and we're exposing it to low CO2, 100 parts per million. And at low CO2, stomata open up. And when they open up, guess what happens? Water transpires and the leaf gets a little cooler. And that's what you're seeing here. If you elevate the CO2, the stomata close and less water evaporating, and the plant gets warmer. So we could, for example, say, well, let's screen at 100 parts per million for plants that don't cool off. And that's what we did. And we isolated a first mutant on this called high temperature 1. It doesn't seem to care what the CO2 concentration is. It's always more closed. And this turns out to be a protein kinase that is a negative regulator. And we're doing really exciting research on this. You know, another question was, well, if high CO2 closes stomatal pores, and I haven't told you really yet, but drought also does. We're going to get to that. If high CO2 closes stomatal pores, there must be CO2 binding proteins, right? That bind CO2 and somehow respond. We don't know the CO2 sensors. And so we decided to look at a textbook and say, well, what kind of proteins bind CO2? Maybe not only a textbook, but basically scour the internet and so on. And we had a list of some proteins that would most likely be in plants and could bind CO2. So then we're like, well, we also in my lab have done guard cell transcriptomes. So we've done cell-specific transcriptomes for guard cells. And I hope maybe I didn't quite introduce what guard cells are. So I'm going back. These are the guard cells, one cell, the second cell, and they can close the pore at high CO2 and open the pore at low CO2. Oh, did I now freeze this thing? Or is it gonna continue on me? Okay, so we've done guard cell specific transcriptomes because let's say we came up with maybe 5 different classes of proteins that could bind CO2. That's not too bad. But in plants, gene families are very large, and deciding what the right genes are, if you have a very large gene family, rather than 5 genes, we're looking at over 60 genes. But because the guard cells respond to CO2, we could use guard cell specific transcriptomes and ask which of these 60 genes are expressed in guard cells. And then we started knocking them out. And we finally found that if we knock out a double mutant and 2 carbonic anhydrases, those are CO2 binding and catalyzing enzymes, and a double mutant, we got a very weak CO2 response in the plant. So let's look at the wild type response first. And when we, so here at this CO2 concentration that's closer to what it is in the atmosphere, if we elevate CO2, stomata close quite rapidly in 30 minutes. And at low CO2, they reopen. And this mutant had a more sluggish response. And it turns out this is a very important mutant. This was originally, we discovered this in Arabidopsis, but it's now been shown to be important in corn and rice. And then you might say, well, who cares? Well, we decided, okay, we have a CO2 binding protein and we have guard cell promoter. Let's overexpress this in guard cells and see what we get. And I'm not going to explain all the data, but the wild type traces are from wild type plants shown here in open symbols. And for our 4 different overexpression lines, we shifted the curve downward, right? Now, without going through the details, let me explain to you what this downward shift means. It means that for every carbon fixed by the leaf by photosynthesis, rather than losing 300 water molecules, the plants on average were losing 40 to 45% less water. And why would that be? Well, I already told you we have 50% higher CO2 in the atmosphere. And we, in this case, are exploiting that by narrowing the stomata. They're more sensitive to CO2. And now for every carbon taken in, they're losing less water. So actually, this is being pursued by industry now to see if they can improve what's called water use efficiency of crops. And the short-term idea is more crop per drop of water. Now, anyway, we did this research in Arabidopsis that's shown here. I told you that that particular mechanism and gene is already found to be important in corn and rice. And corn and rice have different shaped stomata because they're grasses. And they have very interesting morphology there. And we've done a similar screen there recently. So this is now, we're exposing these plants to high CO2, 900 parts per million. In the wild type, the stomata close and the leaves get warmer. This is a mutant, we call them chill because it's cooler. It's not closing its stomata well. And because of that, the leaves are cooler. And you know, you can look at the average temperature here. It's only about not even a 1.5, 2 degrees shift. But with this camera, you can pick up, you can grow 100 plants and ask which one is the mutant, which one looks cooler with a thermal imaging camera. It's pretty cool. And undergrads in my lab use these cameras all the time. Here's another mutant that is also not quite as strong as chill one, but we're also mapping. I'm going to tell you about mapping what the mutant genes are, because that's where a lot of informatics comes in. Okay, this is just some of the biology. I think I'm going to skip this. We want to understand the CHIL1 mutant gene. Why is this not responding to CO2 in contrast to the wild type? Wow. Let me see. Is it? It's frozen again. I wonder if I can. Wow, now it's gone a few slides, or what's that? Okay, yeah. So this slide tells us a little bit about how we can map these mutant genes, okay? So if we have a mutant gene, how are we gonna map it? Okay, if maybe just listen, then I'll explain this in a second. What we do is we take this mutant and cross it back to the parent, and then we have, in the F2 generation, we have a segregation where, in theory, if it's a single recessive locus, what percent of our F2 backcross population should show the CHIL phenotype, the cooler leaves? Just sort of throwing this out there for one of you to unmute and answer. Anybody? So you have a mutant, you cross it into the parent, which doesn't have the mutation. It's a diploid species. You isolate F1 population yourself, and you get the F2. If it's a recessive mutation, what percentage would show the phenotype? You can guess.
0%.
In the F1, it would be 0 if it's recessive, but then in the next generation of selfing,
Oh, 25%?
25%, right. So anyway, so we did this, we collected over 1,000 seeds, we grew about 600 plants. And if we grow 600 plants, and now with our camera, we can select of the 600, in theory, 150 that show the cool leaf phenotype, and that worked pretty well. So now comes sort of where informatics comes in. So the first thing, well, yeah. Anyway, I think I'm down here. I'm going to get back up here in a second. So we cross these and we select, say, 150 that look like the mutant, and maybe 100 or 150, or actually 450 that look like the wild type. And now we isolate DNA from these single 150 plants as well as the wild type looking plants, and we separate them. And for these 150 plants that have been crossed, they could have all kind of random mix of mutations, but for this CHIL mutant, they should be homozygous right there. So we take the DNA from these 150 plants, we pool it, and we sequence that and compare it to the wild type genome. And when you do this, most of this should be kind of random, but at the site of the CHIL gene that causes the mutant, you should see a strong accumulation of that mutant in the CHIL population, but not in the other population. So this is actually whole genome sequence analysis with informatics. We've developed a pipeline for this. And so again, that's sort of what I've written here, wait, actually, I kind of feel like I have the wrong slide here. This is weird. This looks more like for a different project. Anyway, that's weird. I have kind of not the best slide here. So, but so we do informatic analysis to look for that mutation in our short reads of whole genome sequences and maybe long reads to see which regions show an accumulation of mutations. Now, when we compare that to the wild type informatically, we get candidate mutants. It's usually not only one gene, because the crossing over events will give you a few neighboring genes as well. So you might, like in this one case for CHIL1, we have 4 candidate genes. So what do we do next? We also have RNA-seq data of our mutant. I think many of you are familiar with the term RNA-seq and transcriptomes. So we have gene expression data, and we might look at the gene expression data and look at the 4 candidate genes, and maybe we'll see that one of them is expressed much lower in the mutant than in the wild type. That would further help with this. And then as a last step, we might say, well, let's see if we've identified the right gene, so we might knock it out by CRISPR-Cas. Now, we have developed a pipeline to analyze these data sets that uses a number of different software scripts and to analyze these data. And this is kind of an example of something that we train and students in that are interested in this. And it's actually extremely important for our research. Okay, I'm going to mention another thing, and that is if plants experience drought, they synthesize a hormone called abscisic acid. So, and one of the things that abscisic acid does is it also closes stomata. So if a plant is experiencing drought, either the leaves start wilting if it has a weak hormone response, but a good drought-resistant plant produces the hormone abscisic acid, and that's a signal to close stomata. Now, a number of years ago, we were able to, in collaboration with a lab in Riverside, identify the receptors for this hormone, and a pathway was developed for the hormone. How did we identify these receptors? You might just say we screened for a mutant, but that didn't give any receptor mutations. Why is that? Plant genomes have very large gene families. And so you screen a randomly mutagenized population, you will probably not find your mutation because there are a number of other homologous genes that cover for it. So you need a special type of screen. So the way we did this in our screen is we knew that this protein, this PP2C, which is a protein phosphatase, is important very far upstream in ABA signaling. So now we use a technique called proteomics. Have you guys heard of proteomics? Anybody?
Nope.
Nope. Okay.
Yeah.
Oh yeah. Okay. Yeah. Well, I'll still explain it. I guess for others, it's fairly quiet. So these protein phosphatases, we tagged the protein phosphatase, we expressed it in the plant, and then we pull down this with a tag. We isolate the phosphatase and ask what else is attached to it, what else is interacting. And that's how our lab discovered these ABA receptors. And we came up with a model of how the receptors work, which is fine. I'm not going to go into details. But we also started thinking, we should be able to identify more drought-tolerant plants. So actually, we found, like, this is a single gene mutation of an Arabidopsis plant. And these plants have not been watered for 26 days. Okay, something's. Do you guys see my pointer or not really? Is it frozen?
I think it's frozen.
Yeah, it's frozen in your picture, not in mine. I guess pointer is not.
It just moved a little.
Yeah, right. I could see that. Anyway, these plants have not been watered for 26 days, and you can see how the wild type leaves have shriveled. But if you look at the green here, these plants are hypersensitive to ABA. And by doing that, they're more drought tolerant. So this is actually, there was actually a startup company based on this discovery. Can we use this gene in crop plants? And in brief, what it is when you have a drought response pathway, you have positive regulators that close the stomata. But in biology, you always also have negative regulators. It's like in cancer research. You have oncogenes that can produce cancer, but you also have tumor suppressor genes that suppress tumor growth. If you mutate tumor suppressor genes, you can get tumor growth. And if you get gain of function dominant active oncogenes, you can also get tumor growth, more or less simplified. I'm sure some of you are learning that in a more sophisticated way. But similarly in plants, you have, in this case, we mutated a negative regulator of the stress response pathway. And by deleting that gene, we get actually more drought tolerant plant. That was the first time that was actually shown for any plant that you could make a single point mutation and have it be more drought tolerant. Now at that time, like news reporters would call me and they'd be like, oh my God, I think my mom's roses have a problem with your gene because they're wilting or something. And I said, well, this is one negative regulator in a pathway. I think there are a whole lot more than just this one. It's just we discovered a first one. And then the reporter said, well, I guess then it's not that interesting if there's a whole lot more. But it is interesting, obviously. So anyway, so here's a problem with plants. And this is where bioinformatics comes in really big, and systems biology. In order to identify important genes in a pathway that's not well known, whether it's in Drosophila or Arabidopsis, the plant we work with a lot, or in C. elegans or mice, what's a good way to identify important genes for a process? You do a genetic screen. You screen for mutants, right? Well, and that's been really powerful in plants, but one limitation has been that because gene families in plant genomes are much larger than in other organisms, which has been found. If you mute, you're not going to find it that easy through a genetic screen. So what that means is, even though a lot of scientists have used this plant, Arabidopsis, for genetic screens, through loss of function mutations, only about 12% of the genome have been identified to have a loss of function phenotype. So we were like, and the reason is you can't just mutate one gene. You have to mutate 2 or 3 or 4 homologous genes, and then you get the phenotype. So most of the genes in the plant genomes are hidden in that way and not easy to find. So what we did, and when Felix, oh boy, is this not working again? Diego, did you ever get my email? Because this is kind of boring here.
Yeah, I have it downloaded. I can try to share.
Let me see if I can, if someone can stop me from sharing, that would be nice. I don't know if I can do that. Touch screen. Yeah, I could do it even without a mouse because I have a touch screen. And let's see if you can do that.
Can someone give me access to share, please?
Yeah, that might be.
Yeah, I'm doing it right now.
Okay. Yeah. Wow.
Okay, so you should be able to share.
Okay. All righty. Okay, so the idea was the following. Let me see. I have the screen here now. Wait. Yeah, I have the screen here and my camera's up there. So I'm gonna, wow, there's something weird here. Let's see. Yeah, I'm gonna just swap around a few things. Okay, here we go. There we are. Actually, no, wait. What am I? I'm seeing my slides still. That's weird. What happened here? This is so weird. Why am I not seeing Diego's? Wait. Huh. Why am I'm seeing my slide and not yours. Something's mixed up here. Maybe because it froze on that. And it was there a second ago. I have 2 screens. Maybe I should unplug one of my screens.
Can you guys see the screens I was sharing?
Yes.
Yeah.
So weird. I see my screen and I don't see yours. I'm gonna leave and log back in, okay? That's so strange.
All right. Well, while he does that, I guess I'll just take this moment to say that if you guys are interested in the bioinformatic aspect of these analyses, which he doesn't go into too much detail here, since he's focused more on the biological relevance, feel free to, I guess, contact me.
Yeah.
If you have any.
So Diego,
Okay.
I thought I was still on my slide because I thought we were going to see the next slide. So maybe you can go to the next slide. Yeah, there we go. So maybe it wasn't frozen, but maybe it's just that I thought we were already on this next slide. And then the other slide popped up. Okay, so plants have very large gene families, and we decided we want to develop a technique to knock down homologous genes in different combinations. And so the first thing we did that's shown on the left is and Felix Hauser in my lab spearheaded this. We said, let's develop microRNAs that don't silence one gene, but have nucleotide sequences so they target close homologs, but no offset genes. And we did this on a genome-wide scale for gene families. And you can do various combinations. So we actually got access to the Triton cluster at the Supercomputer Center. And iteratively, we used 90,000 hours of their computation time on their 2048 parallel CPUs to design microRNAs that would co-silence genes, okay? And maybe in the next, and okay, on this Phantom database that's listed on the bottom of the slide, we list, there are actually 2 million microRNAs that we designed computationally that could silence different combinations of the genome, silencing of the genome. But in the next slide, let me show you an example. So what you see here in red are 5 different genes, okay? And what you see in green are microRNAs that targets different combinations of them. So if you look at the top left microRNA, it targets the 2 red genes there. But microRNAs silence genes. They don't always do it perfectly. So we have another microRNA to the right of that that silences those 2 and another gene. So really what we've designed is sort of a combinatorial gene space of silencing different combinations of closely homologous genes. And so we started doing genetic screens with this in the next slide. And if you look at the very bottom of the slide where it says pill 4, pill 6, peer 1 on the bottom right, those are the ABA receptors. People tried to discover the ABA receptors for decades. We finally found them 10 years ago. And actually Science Magazine had then called that, I think, the 2nd top breakthrough of the year or something like that, because of drought tolerance and its importance. With this screen, we immediately found these genes like that. And the green dots here are microRNAs, and the red dots are different combinations of genes. So we immediately rediscovered known genes, but we've also discovered major unknown genes. And you know, how do you do a high throughput screen if you have all these microRNAs expressing in plants? So each plant expresses 1 microRNA. And we screened something like on the order of, let me think here for a second. Yeah, okay, yeah, roughly 1 million different seeds. Well, how do you screen 1 million different plants? In the next slide, I'll show you how. This hormone ABA, as you can see on the bottom left, inhibits germination of seeds. So you can screen 1 million seeds on a plate and you just see these dots on a plate. But if anything germinates in the presence of ABA, as shown to the right of there, that's probably a mutant. Now in this case, we're doing repeat experiments of 1 microRNA expressing line. So this is how we would screen for mutants. And in this case, we published a paper last year where we discovered a very important mechanism that plays a role in drought tolerance. And I'm not going to go into the details, but maybe in the next slide, we discovered these MAPKKs here that were kind of a missing link in this pathway. And actually, you know, considering that we just published a paper, it's already drawn a lot of interest in it. In my last part of the talk, I want to mention one other example. So I've mentioned to you a few examples where we're using bioinformatics to address our research. And as I said earlier, we have really cool projects. If you're interested as an undergrad in this and you're not graduating in June or whatever, you know, then let me know, because it might be a little short right now between now and June. but we certainly have interesting research. I'm going to mention one more project in the next slide that Diego, among other projects, Diego has been very involved in. So we're part of the UCSD Superfund Center. What is that? Well, it's a big grant. It's like a $15 million grant or something like that. And we just submitted a renewal. And a lot of the scientists in this grant are in the medical school, and I'm the one in the biology division that's in it. And it has to do with the fact that spread across the United States, there are these sites that are known to be contaminated. Many of them because of early industrial revolution, not taking care of where you spill your toxicants. And they're called Superfund sites because they're very expensive to clean up. And the top, there's a list of over 200 compounds that are toxic at these sites. But as you can see on the right, the priority substances, the ones shown in purple are metals like lead, mercury, cadmium, or the metalloid arsenic. And to remove those, it turns out plants are really good at removing metals and cleaning up soils for bioremediation. So the next slide, we have a collaboration with the University of Arizona. And let me explain this. So there are contaminated sites, for example, in Arizona, that are these old mining sites, and they're just huge areas of contaminated metals in the soil. You can't grow a normal plant in those soils. And there's dust and wind, they're dry. So dust blows up and it blows into cities, whether it's Phoenix or some smaller community nearby, and it's toxic. It's toxic if you inhale it. It's toxic if it lands on your foods or if it lands on the soil in which you grow foods. So we're not growing foods here, but we're growing plants that are good at removing those toxicants there. And this is a plant that Diego has also worked with, quail bush, on the informatics end. And we're asking why and how are these plants able to grow in these highly toxic metal mining sites? It's really an important question. And this is a plant, quail bush are also called Atriplex lentiformis, that has not, for which the genome had not been sequenced, but we did transcriptome analyses to ask which genes are expressed, which are induced by metal stress. And we're doing that, that's still ongoing. And maybe next slide. You know, how do these phytostabilizer plants, as we show them, do bioremediation? How do they grow on these highly toxic soils? So Diego actually has been doing transcriptome analyses. Now, in this case, this is a plant for which there's not an assembled genome. So what we do is we do RNA-seq and we do something called de novo analysis of this by comparing this to other genomes. Are any of you familiar with de novo assembly of a genome? If you have, say, transcriptome data or genome sequencing data from short reads. Any of you guys familiar with de novo sequence analysis? So that means if you don't have a sequenced genome, you can still use short reads and algorithms to try to assemble a genome or transcripts and ask how high are they expressed. And just in the next slide, this comes from some of that analysis. It's just an example. It's ongoing analysis. So what we see here, for example, on the top, you see on the top are selected genes that are called to be expressed very highly. Let me get this right, in a certain situation, but lower, red in the other. And on the bottom are clusters of genes that are expressed more highly on the left and lower on the right because of our use of the red, and you can see the color key on the top. So we're basically asking what kind of genes allow these plants to grow in these very toxic environments. And that's kind of important if you want to secure these Superfund sites. And like I said, there's a major project at UC San Diego, which has to deal with how do humans, how do animals, how does nature, and how do plants deal with toxicants that are available in these sites. And that's not only of interest to the United States. It's also of interest to other countries where you have such toxic sites. So in the next slide, and I'm getting close to the end here, so how do we do these analyses? Well, I mentioned we do RNA-seq analyses. of plant tissue grown on different toxic soils. We do transcriptome data sets via de novo genome assembly and analyses. We use differential gene expression programs like DSeq2 and so on, but there's a lot of sophisticated analyses that go into that. That's, well, sophisticated, it's fun and you can learn it. And we do comparison. We're actually planning, this is a future thing. We have one species, this Atriplex that we're analyzing now that is very good at this. And then we have a very closely related species from the same genus that does a different thing with the metals. So we want to compare those. That's kind of a future project. And maybe you can just go to the next slide to see what's there. Yeah, I think I'll leave this here, leave this out, but we have other such examples. Yeah, okay, I'm just going to mention this one here. Another example where we do a lot of projects linked to bioinformatics. So we're interested in how plants withstand drought stress, and this ABA is the drought resistance hormone, and we're looking at chromatin modification in response to ABA by doing attack-seq and RNA-seq comparisons and things like that. So there's a lot of exciting informatic analyses. And in the next slide, I'm just kind of listing here and thanking people in the lab who contributed to this. And I'll just mention some of the names here. Chuck Seller is in the lab right now, and he's doing attack-seq type experiments to look at chromatin modification in response to stress in plants. That's probably extremely important and hasn't really been possible to analyze before. Felix Hauser developed this artificial microRNA library. I'm jumping down. So Sunny Wang and Diego are both bioinformatics students at UCSD, and they're helping with a lot of these analyses and actually doing huge analyses. So very likely they will be ending up on at least one, if not several publications based on their research. Alexandria Tran was a bioinformatics student. Maybe some of you know her. She graduated last summer or in June last year and is now a PhD student working on, actually she decided to work on plants. She's working on systems biology of boosting nitrogen fixation, which is very exciting and important. And I think I'll leave it at that. And I'm certainly open to discussion. I did put my email address up here so we can, you know, maybe leave the slide up a little longer. If you want to contact me, it's right there. I've been told it's not so easy to find, but it's, I mean, you can find it. I don't know if that's true or not. I haven't really checked. But if I'm looking for someone's email address, I don't find it on their website. I go to their publications, and there it is, communicating author or something like that. But anyway, that's just a tip for the future. So do you guys have any questions? And sorry about the technical difficulties. I'm gonna have to figure out what that is, because I kind of noticed it yesterday when I was playing with this thing. When I put the talk on Zoom on my computer, it doesn't cause the problem, but on Zoom, I don't know what's going on. Anyway, any questions?
Is there a trade-off to drought resistance? It seems like there's a lot of benefit to it. You lower your water input, but then what's the trade-off?
Right, so the trade-off is you don't want to have a plant that's always drought resistant. And the reason for that is, let's say, for example, you close your stomata. If you're closing your stomata, you can't grow because you're not taking in as much carbon. So you want the plant, when there is water, to open its stomata, but when there's not, to close its stomata. The other thing, the hormone ABA doesn't only close stomata, it inhibits seed germination. How does a plant survive drought? Well, short of improving water use efficiency, and we have new methods to do that with our CO2 response, short of that, ABA is a hormone that slows down growth, because that's a good way to survive. But if you're trying to grow a tree for certain purposes, or certainly a crop plant, you want it to grow when water's there. So you want to amplify the ABA response under drought conditions, but then you want to be able to turn it off again. And the reason the CO2 research I mentioned to you is very important is, even if there's not drought, we can, based on the CO2 concentration in the atmosphere, narrow stomatal pores a little bit, so they lose less water while they're still taking in the same amount of carbon because there's more CO2 in the atmosphere. So that's a really excellent question. So, you know, another thing that's interesting in terms of informatics And synthetic biology is developing drought-inducible promoters so that only under drought do you have maybe a more stronger drought resistance. So there's a lot of interesting subjects here. Yeah. Any other questions?
Okay, so I guess if nobody has any other questions, we'll end it there.
Okay.
So thank
Well,
you.
yep. Thank you all very much.
Thank you, Dr.
And,
Schroeder.
If anybody wants to hang around, I don't know if this is gonna shut off when you go off, but if anybody
I
has any
think
more
since
questions,
you're co-host, I can just leave actually.
yeah.
So if
So,
anyone, any of you
yeah,
want
if
to-
anybody wants to stay on, I'll just hang around.
Okay, sounds good. Bye.